Hey everyone, this is episode 652 of the People Behind the Science podcast. This week we are rebroadcasting our interview with Dr. Franck Polleux, originally published on March 14th of 2016. Listeners, Franck is an outstanding cellular and molecular neuroscientist, and he is one of the first scientists to explore how human-specific genetic changes may impact the development and function of key circuits in the brain that underlie cognition, a process that could help explain the extraordinary capacity humans have for complex thinking. And in our conversation, Franck shares more about his career, his life, and his research. So listeners, get ready to learn more in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am delighted to be here today with Dr. Franck Polleux. So, Franck, welcome to our program. How are you today?
Thank you, Mary. I'm doing great, and I'm very happy to be here today.
Well, we are excited to have you on the line with us, and I'm excited to introduce you to our listeners. So I'm going to start by giving them just a little bit of background information here. Now, listeners, Franck is a professor of neuroscience and member of the Zuckerman Mind, Brain, and Behavior Institute at Columbia University. He completed his undergraduate degree, as well as his PhD in neuroscience, at the University of Claude Bernard in Lyon, France. Afterward, he traveled to Johns Hopkins University for his postdoctoral fellowship. And Franck served on the faculty at the University of North Carolina, Chapel Hill, as well as the Scripps Research Institute in La Jolla, California, before joining the faculty at Columbia, where he is today. And Franck is here with us today to talk about his journey through life and science. So, Franck, we are going to ask you lots of questions, and we really want to get to know you, both as a scientist, but also as a person. So can you start by giving us a little window into your life outside of science and how you spend your time?
I enjoy outdoor activities. I love biking, and believe it or not, I can actually enjoy biking in New York. It's actually much easier than people would think.
Really? It's quite an obstacle course, I think.
Yeah. I also enjoy other sports like tennis and basketball. And I also have two small kids, 8 and 11. And so I spend as much time as I can with them doing silly things and going to theater and read with them and things like that.
That sounds fantastic. So are your kids getting into things like tennis and basketball and biking too?
Absolutely. Especially biking. They're really into biking.
Awesome. Well, great to get a look at your life outside of science, Franck. But next, I do want to talk about your scientific side. So can you tell us how you describe what you do to someone who might not be familiar with science or your field?
My lab studies the cellular and molecular mechanisms that underlie brain wiring during development. That's really the primary focus of my lab. We're interested in aspects of brain wiring, such as how neurons form dendrites and axons, how these axons and dendrites grow and branch in a very patterned way, and finally, how axons make synapses onto the dendrites of other neurons, basically. We exclusively use the mouse as a model, mostly because of the genetic tools that are available in mice, and also because it's a mammal. And it has a relatively small brain, but its structure and wiring diagrams is very similar to other mammals, including non-human primates and humans. We're also interested in how neuronal connectivity might be different in the human brain compared to other mammals. I'll talk to you later about this in more details. And we're specifically searching for human-specific genes that could act as modifiers of brain development. And finally, more recently, my lab also started studying some of the signaling pathways that are altered in early stages of neurodegenerative diseases, such as Alzheimer's disease. It turns out that some of the signaling pathways that we study during the development of brain wiring, basically, seems to also to play a role in maintaining those connections during neurodegenerative disease. So those are the three main interests in my lab.
Well, Franck, those were great little teasers of those three main areas in your laboratory. And our listeners know this by now. I love talking about the brain. So I'm looking forward to chatting more about your work later on in our show. But I want to talk next about what motivates you. I think as a scientist, there are a lot of hurdles to overcome. There are a lot of challenges. There's just a lot to get done in the day. So it always helps me when I have a little quote or a saying, something that I can keep in my mind to keep me going. So Franck, do you have a favorite motivational or inspirational quote?
Yes, I do, actually. One of my favorites is one that is usually attributed to Winston Churchill. I think he used it many times, and there are many different versions of it, but basically boils down to the following. Success consists of going from failure to failure without loss of enthusiasm. I really like this quote. I think it's particularly relevant to the scientific process. As scientists, as you mentioned, we're constantly facing failures. You design experiments to test an idea or hypothesis. You perform the experiments, and most of the time, those experiments don't work, either for technical reasons or because you didn't design it in a way that optimizes your chances of detecting what you're looking for. And sometimes the results are very clear and your experiment works, but they show exactly the opposite of what you predicted or hypothesized. Mm-hmm. And there are many sources of failure and disappointment in the scientific process. I think that part of what makes successful scientists is to be able to go through this process without losing enthusiasm and passion for the question you're tackling. It's clearly not easy. The general public probably usually don't realize that science is hard and challenging. You need tenacity above all. And it's challenging and frustrating at times. But it's also a lot of fun, of course, when it works. And when you discover something new, it usually makes up for the multiple failures you had to circumvent. I still consider myself very lucky to be able to be a scientist. But I love that quote because I think it sums up what science is about.
Absolutely. And I love what you said here about when those successes happen, when you make those discoveries, it's sort of much easier to forget all those failures that came before it. But
absolutely.
So would you consider yourself one of these optimistic, enthusiastic people, generally speaking, or is this something that you really have to dig out when you're in one of these tough times?
No, I think I am an optimistic, sometimes in a very naive way. I am in general very enthusiastic. And that's something over the years that I noticed and that people around me, my coworkers, my friends, point as a quality. I hope it is one, but I think it's a very important quality in science.
Certainly. And I'm sure one of these things that your mentees are trying to emulate, because this is something that's hard sometimes, particularly in these long stretches, these long dry spells in science. So great to hear this motivational quote that keeps you going. But I want to chat next about some of the people who've helped kept you going along your journey in science. So can you tell us, Franck, about some of these role models or these mentors who you've had and really looked up to?
I, of course, had fantastic mentors, both during my PhD and my graduate work and my postdoc, and even after that. I don't think they're really role model in the sense that admiring someone as a scientist to the point of wanting to become like him or her. However, I think there are many scientists that profoundly influenced me, and we'll talk more about this later, but I was fascinated by two French biologists, Jacques Monod and François Jacob. They both won the Nobel Prize in 1965 for their pioneering work in the 50s and 60s on identifying key principles regulating gene expression in bacteria. Their work had a profound influence on me. But on top of those extraordinary scientific achievements, their persona, their courage, their life history in the resistance during the Second World War were almost legendary to me. I admired them both as humans and as scientists very much.
Well, this sounds fantastic. I love having these historical figures, these big mentors in mind, as exactly what you're saying. You don't necessarily want to become them, but these people to look up to kind of take little bits and pieces of the things that they've done well over the course of their career and trying to emulate them yourself. So fantastic, Franck. And we've hinted a little bit at little pieces of your journey and how these people might have inspired you along your journey. So let's talk about your journey next. Can you take us to the beginning and tell us what first sparked your interest in science or the brain?
My interest in science in general, but as a neuroscientist, really came about fairly late, actually. During college, basically, right before I decided to do a PhD in neuroscience. I come from a pretty unusual background in the sense that no one in my family is a scientist or even expressed scientific interest when I grew up. Mm-hmm. They're all educated. For example, I have one of my aunts who lives in Paris. Jeanne Faya is a writer, but she writes about artists and sculpture and things like that. My parents come from the north part of France, and after the Second World War, the 50s and 60s, they worked in the textile industry. They moved to the southern part of France in the Alps, where I was born, and then to Lyon, which is the second largest city in France after Paris. And at that time, they changed jobs and worked for the rest of their life as bank employees, basically. So no one close to me in my family was a scientist. And I spent most of my life in Lyon, where I studied all the way to college, and I did my PhD in neuroscience there too. I did my PhD with Henry Kennedy and Colette Dehaye, working on the relationship between neuronal birth dates and the specification of neuronal fate I was studying basically using fairly quote unquote old fashioned methods, the relationship between when neurons are born and the position they occupy in the brain, and how this birth dates and or their position dictated basically where those neurons projected in the brain.
Very cool. I love this idea of a neuronal birth date.
Yeah, no, exactly. It's a magic neuron with a little candle there. Right. But it's a fascinating story. And I actually learned tremendously from both Henry and Colette during this process. Anyway, then I moved to the US for my postdoc at Johns Hopkins in Baltimore, where I joined the lab of Anirban Ghosh in the Department of Neuroscience, which was and still is a fabulous environment to do neuroscience. I learned at that time more about cellular and molecular techniques, and I started working on identifying molecules and principles that pattern and guide axons and dendrites in the cortex. And then I moved back to France for 2 years, actually, after my postdoc, but it didn't really work out and as I expected for many reasons. It was difficult at that time, this was early 2000, to really become independent quickly, basically, in the French system. And I decided to move back to the US to start my independent career. And I got my first job at UNC Chapel Hill, as you mentioned, where I worked for about 8 years. I really loved it, both personally and professionally. I owe a lot to this period. I think it convinced me basically that I could become an independent investigator and have my own lab. And then, as you mentioned, in 2010, I had a job offer at Scripps Research Institute in La Jolla, in California, which I also enjoyed tremendously. And I thought that I would stay there for a while. But in 2013, I was approached by Tom Jessel, who's a professor here at Columbia, actually one of the neuroscientists that I considered a role model, or at least that I admired and who influenced me tremendously. And he approached me to join Columbia University, to join this new institute called the Zuckerman Mind Brain Behavior Institute that will officially open next year in a new building on a new campus at Columbia. And the breadth and ambition of the project itself of this institute dedicated to study neuroscience and the brain was very attractive to me. And I thought that this was a once in a lifetime opportunity to join one of the best neuroscience community in the US. And I decided to move to New York, which I actually also enjoyed very much as a city. It's been 2 years now, and it has met all my scientific expectations so far.
Well, that is fantastic. It sounds like you have had a wonderful journey through science and that you're really enjoying the place where you are today. I'm curious, you mentioned that you didn't have any family members in science and you didn't really know that this was necessarily the career that you wanted to pursue. And I was in a similar boat. And when I went to college, this was kind of the first time that I ever thought like, oh, this is something people can do as a career. So do you remember maybe one of the first experiences that you had that really started you thinking about research being your own career?
Yeah, very much so. In France, unfortunately, we don't have a rotation system before you decide to join a lab to do your graduate work. You essentially, quote unquote, left alone, try to decide and identify your lab.
Right. Blind choice. It's like a blind date.
Exactly. But you do that through working for summer, for example, doing internships in labs and things like that. And so I worked briefly at the end of my fourth year in college in the lab that I eventually joined, the lab of Henry Kennedy. And I was just hooked. I realized that both the environment was incredibly stimulating and the topic was very interesting to me, just studying brain development. And this was obviously the right choice for me. I really enjoyed it.
Fantastic. Now, we mentioned the Mind Brain Behavior Institute at Columbia, where you are today. I want to hear a little bit more about this place now. And can you tell us about one of these projects you have going on there that you are just the most excited about, Franck?
Yes, so the Zuckerman Mind Brain Behavior Institute will house a large number of labs at Columbia, about 50 labs probably in total. The goal is to tackle brain development and brain function through very multidisciplinary approaches. So we're trying to gather a number of people that tackle the brain through imaging approaches or genetic approaches or in various model organisms. So it's going to be a powerhouse, I think. I'm very excited to join the Institute. I think one of the most exciting and daring projects in the lab currently revolves around a central question, which is what makes us human? And more precisely, what makes our brain human specific? It's a big question, of course. It's a question that has fascinated scientists and philosophers for centuries, really. but we still know very little about what is so special about the human brain that allowed the emergence during evolution of our rather unique cognitive abilities, such as artistic creativity and abstract thinking, or the ability to communicate through complex forms of languages. Mm-hmm. This is, of course, a very complex question, and it has always fascinated me for many years, but I never really quite knew how to tackle it. This was really more of a hobby, I would say. But about 6 years ago, I decided to tackle this problem and take a genetic approach to this, to study the role of human-specific gene duplication and the role of human-specific gene duplication in brain development. So just to back up a little bit, gene duplication is a major driver of evolution and speciation. Some genes get duplicated through the process of meiotic recombination. And then the new copy of that gene can acquire a new function through specific modifications. For example, it can be mutated or truncated. And it acquires a function that's usually different from the ancestral copy. And if this new copy basically provides a selective advantage, then it might get selected in subsequent generations. So that's really the process of speciation.
Gotcha.
So work in several labs over the past 8 to 10 years have identified a number of genes, a large number of genes, relatively approximately 35, 40 genes, that have an extra copy only in the human genome. In essence, this is the most evolutionary recent form of genomic evolution that is specific to the human genome. So these duplications are not found in any other mammals, including non-human primates, right? They're really human-specific. Unfortunately, or fortunately, none of these genes have any known function, and especially not in the brain, but most of those genes are expressed in the brain. That's actually very interesting in and by itself. With one exception, a gene called SRGAP2, which my lab was studying when this first paper reporting that this gene was specifically duplicated in humans came out. So we discovered that the ancestral form of this gene, the copy of the gene that's shared among all mammals, in fact, this gene is much more ancestral than mammals. It's present in C. elegans and Drosophila, for example. But we discovered that the ancestral form of this gene plays many functions during brain development. In particular, it's a critical regulator of synaptic development, the development of synapses that constitute the junctions that neurons use to communicate. This gene, in particular, SRGAP2, the ancestral form of this gene, limits the density of both excitatory and inhibitory synapses that a neuron receives. And it also speeds up the maturation of those synapses during development.
Interesting.
So basically, when we knock out this gene in mice, when we inactivate the ancestral copy of this gene, neurons receive 30 or 40% more synapses, and these synapses mature at a much slower pace. And so we discovered that the human-specific duplicate of this gene is a partial duplication, which is very often the case, actually. When genes get copied, very often they get truncated or they get altered. And this gene is expressed in the human brain. We can detect a messenger RNA, we can detect a protein, and it gives rise to a protein that's truncated by about 2/3. But the third that's remaining retains the ability to bind to the ancestral copy of the protein, and it largely inhibits its function. So this was an important discovery in our research. And what we did then is when we expressed this human-specific gene duplication in mouse cortical neurons, we obtained the same phenotype that we get when we knock out the ancestral copy of the gene. Similar phenotypes. Mm-hmm. Essentially, neurons form many more synapses, and these synapses mature at a much slower pace. So we published a paper a couple of years ago reporting these first findings, and we were very excited about this because this relatively simple gene duplication event allows for the emergence of two properties that we know are different about human neurons compared to other mammals. Human neurons are hyperconnected. Studies over the years have shown that they do receive more synapses than the equivalent neurons in non-human primates or other mammals. And we also know that synaptic development is much slower in humans, a phenomenon that usually people call neoteny, which refers to retention of immature features for longer periods of time. Mm-hmm. And so basically just implementing this single gene duplication in mouse neurons seems sufficient to humanize two key features of human neurons, i.e. form more synapses, and those synapses develop more slowly. So these results raise many more questions than they answer.
Right. As usually with the exciting results. Yeah.
That's right. The first one is, what is the consequence of implementing these changes, right? This increased number of synapses and synapses maturing more slowly for circuit function and behavioral performance, right? We don't know yet. it is actually a central question in neuroscience. What is the relationship between the degree of connectivity between neurons in a circuit and the ability of this circuit to compute information? And the second is, what is the relationship between circuit function and behavioral performance? So theoretical neuroscientists have predicted that if you increase the number of connections in artificial neural circuit, you increase the capacity of this circuit to compute information, much like a computer. If you increase the number of microprocessors, for example, your computer can increase its efficiency and speed at doing things like complex calculations, right? Mm-hmm. But for biological neural circuits, we don't really have any experimental evidence that this relationship exists. So we think that our findings on SRGAP2 and its human-specific duplication will allow us to test this question for the first time. We made transgenic mice that express this human-specific copy of SRGAP2, and we're testing this as we speak. And this is one of the reasons, actually, I moved to Colombia, to be honest with you, because many of my colleagues here in the neuroscience department and the Mind-Brain Behavior Institute have developed unique experimental approaches to tackle circuit function in vivo and the relationship between circuit performance and behavior, basically. And so we're very excited because we think we have a unique experimental model in mice to really tackle these complex questions of what makes us human, more precisely, what could be so different about human neural circuits and how those differences might explain maybe higher cognitive abilities. That's really probably one of the most exciting projects in the lab right now.
Well, Franck, this is fascinating work. Thank you for sharing some of the details about this project. And I think you're absolutely right. This is one of these keep you up at night kinds of questions. What makes us human? What makes our brains different from the brains of other mammals? So fantastic question to be tackling in the laboratory. And I'm curious, I know this has, as you mentioned, a lot of philosophical implications as well. Do you dabble in the philosophy of it as well on the side?
Yes, absolutely. And I was fortunate enough to study philosophy in France. This is part of the regular training, basically, up until when you're 18. Philosophy is taught at a pretty high level, actually. And I was especially fascinated by the philosophy of the scientific process and the relationship between experiments and ideas and how ideas evolve as knowledge evolves. And so this is something I'm still fascinated by. Absolutely.
I love it. Well, Franck, I think we've talked about a lot of the great aspects of your current work, your career, and how you got there. But I want to go back to our jumping from failure to failure with undiminished enthusiasm idea and talk about some of these failures. So I know there are so many of them in science. It's hard to pick a favorite sometimes, but I want you to share one of your own failures with us and tell us how you got through this tough time.
So that's a very good question. So I think that probably the most challenging times I had during my relatively short career was during my graduate work, my thesis work. As I mentioned before, I was studying neuronal birth date, and the way we did this was, not to get into much detail, was by essentially labeling neural stem cells as they generate neurons. So that's what we call neuronal birth date, because neurons are fully postmitotic, right? Once they exit the cell cycle, once they're generated, they won't divide anymore again. So there's a clever way to label neuronal stem cells as they're dividing. And when they are generated, essentially when neurons are born, they inherit a fixed amount of this tag, of this labeling that you get. And you can actually estimate when they were born based on when you actually label them during development.
That's cool.
Yeah, it's a cool technique. It's still used heavily. But at that time, the technique we were using to label those cells revolved around incorporating a radioactive compound. And the work I was doing was painstaking. It involved basically counting small dots, basically neurons, and I had to count literally millions of those cells and
oh my gosh.
map their distribution pattern throughout the brain. It was painstaking to a point where I was doubting that I would even finish this work. It was really, really years and years of work before I could even imagine publishing this work. Mm-hmm. But it did turn out very well, and in the end, I had to go through this. But I have to admit that I really benefited tremendously from my mentors at that point, because I think mentors in science are there For the good times, of course, sometimes. But they're there, as I mentioned before, mostly in the bad times, mostly in the times that are really challenging, either intellectually or experimentally. And both Henry and Colette were very supportive throughout this process. And I learned a lesson during these times, which is never give up. I mean, no matter how difficult a project is, if you're convinced that the answers you might get are important, then plow through and persist. I think persistence and tenacity are, again, very important in science.
Well, very well said, Franck. I think a lot of graduate students listening out there might be nodding their heads if they're stuck in one of these insanely tedious processes as part of their research. I know I did a lot of counting myself, either counting neurons, counting zooplankton, things like that. And it is really frustrating because hours into it, everything's starting to swim before your eyes. So how did you, in the moment, get through those challenging, long days, many hours spent just doing something so mundane?
In many different ways. I have a passion for music, and I think music saved my life in many ways. Mm-hmm. One of those ways was just during those very long hours at the microscope, just listening to music. It was very motivational from time to time. It doesn't work for very long. Sometimes you have to do something else.
You alternate between classical and angry music.
Exactly. Between jazz and metal.
Right. Yeah.
So trivial things like this make a big difference. I think, again, my friends and family were also tremendously supportive during that time. My parents have always been incredibly supportive and proud of my achievements. And when things were not going so well, it was critical to have this level of support.
Absolutely. Well, thank you so much for sharing the story. I think having the support of your friends, of your family, of your mentors in particular during these tough times is critical to getting through them. So wonderful story to share with our listeners, but we don't just want to talk about the difficult times. Franck, I want you to tell us about one of these success stories, one of these moments that was really important to you when you look back, whether it was a big victory or even just a small win that was really special.
That's a little more difficult, I think, because it depends what qualifies victory. I think, as you mentioned before, I actually still very much enjoy the little victories, which in the lab consists mostly of people knocking on my door and my students or my postdoctoral fellows in the lab coming and telling me, oh, come look under the microscope.
Right. Excitingly dragging you away.
Those are the moments I cherish the most because as a PI, you basically get bogged down in writing reviews and reviewing articles and writing grants and things that are actually, I personally don't enjoy that much. I enjoy still looking under a microscope or looking at a screen and realizing and observing something new. There's been recently a study that came out of the lab. We published a paper in Cell in 2013 where we had a couple of aha moments during this process. This is pertaining to the other topic in the lab, which has to do with identifying genes and proteins and signaling pathways that regulate the ability of axons to grow and branch in specific patterns and form connections. This particular study revolved around the study of a kinase that's called LKB1. And the ability of this kinase to regulate axon branching and the stability of those branches through a very unique and novel cell biological step, which revolves around capturing mitochondria at presynaptic sites. It sounds very specialized and incongruous. And in fact, it took us 5 years to find this relationship between the ability of axons to branch and within each of those branches, the necessity basically of mitochondria to stop at specific points along those branches, namely at synapses, at sites where communication occurs. I distinctly remember when we realized that there was a connection between this mitochondrial capture at synapses and axon branching. It was very rewarding for the two postdocs who were involved in this work, Julien Cochet and Tommy Lewis. And I have to admit that this was a long journey. This was a challenging one, but a very rewarding one. It spurred many other projects in the lab currently. I have 4 other people right now sort of pursuing this direction. So it generated a lot of enthusiasm and raised many more questions that we're currently tackling.
So, Franck, I have to ask, after this long, arduous journey, you mentioned this paper was published in 2013. How did you celebrate the successful moments with these 2 postdocs who had been slaving away on this project?
There's a couple of ways we celebrate big papers in the lab. The first thing we do is we open a good bottle of champagne. I love champagne. I'm French after all. Mm-hmm. And then we sign that bottle and I proudly display a few of those bottles on a shelf in my office. That's one of the ways we traditionally celebrate new papers in the lab. But also we just go out and have a drink maybe a couple of months after that when things calm down. I think it's important to maintain this level of celebration because it's not a single moment. It takes 5 or 6 years to get to that point. So I think it's also important to keep celebrating those moments with people that are involved.
Well, I absolutely agree. I think all these little wins deserve little celebrations along the way. And maybe when you get to that big milestone, you can crack open a bottle of champagne, go out for a drink, something like that. So
exactly.
Fantastic to hear about one of these successes of yours. And I know earlier in our interview, when we were talking about life outside of science, you mentioned that you like to spend time with your kids and you like to read with your kids. And I love talking books on our program. So I want to chat with you about books that you would recommend. And this could be one that you read with your kids, a science book, a non-science book, whatever you have on your shelf that you'd recommend.
Unfortunately, my kids are a little too small yet to appreciate the 2 books I'm going to mention, but I can guarantee you that at one point I will read them.
Right. Wait a couple of years.
So I can think about 2 books for very different reasons. The first one is a book called, in French, La logique du vivant by François Jacob. It translates into The Logic of Life. Came out in 1970. I read this book when I was 18. Actually, it's very funny. As I was preparing for this interview, I found the original version of the book that I still carry. It's in my hands right now. And I actually had the weird habit of writing the dates when I read those books. And so, yeah, I read it in May 1997.
Oh, wow. That's cool. I'm going to have to start doing that.
Yeah. No, it's great, actually. And this book had a transformative impact on me. The book is sort of a scientific and philosophical account on his journey through science. And he's trying to explain how life emerged and what is the basis for heredity, how life and organisms evolve, how embryos are patterned during development, how genes and protein works in cells. It's a beautiful book. It's a personal account on how microbiology and the emergence of molecular biology revolutionized our understanding of nature, life, and evolution. I was absolutely fascinated by that book. I have to admit that the book itself was not very easy to read for me when I was 18, but it challenged me to learn more and understand every single concept and facts contained in that book. I made a promise to myself that I would slowly or quickly learn everything and understand everything that François Jacob wrote in this book. And it motivated me to go on and study biology in college, actually. So this book had a profound impact on me. And I actually reread this book just a couple of years ago. It's still incredibly modern and it's definitely worth reading.
So have you mastered it all?
There are still some things in there that are more on the philosophical side. It's still a little puzzling.
Have to mull it around a little. Yeah.
That's great. That's what life and science in general are about, right? They still have more to learn. We never stop learning. That's the beauty of being a scientist or being a human, in fact.
Absolutely. Was there a second book that you wanted to talk about?
The second one is called Brave Genius by Sean Carroll. I don't know if you read this.
I have not.
It came out a couple of years ago. Sean Carroll is himself a very incredibly talented biologist and evolutionary biologist. It's a fabulous book about the parallel history of Albert Camus, the philosopher and author, for example, of The Stranger, and Jacques Monod, the biologist I mentioned earlier in the interview. So after a German invasion in France, both men ended up resisting the oppression in many different ways. Monod by joining the resistance and moved to England to join the Liberation Army by de Gaulle, and Camus by becoming a writer and a journalist who denounced German oppression and also denounced the French government of Vichy, which largely collaborated with Germans to impose the rule to French people for 4 years. Both Monod and Camus took incredible risks during this time period, and their acts of resistance were very different in many ways, but amazingly brave. And after the war, both of them became incredibly successful, overcame this adversity, and ended up winning the Nobel Prize for Albert Camus in literature in 1957. And for Monod, together with François Jacob and André Leboeuf, they won the Nobel Prize in medicine in 1965. The book by Carroll is incredibly well written, very well documented. It represents many years of hard work. Carroll mentioned in interviews recently that I saw that this was very much like a detective work with one of his assistants in France, where they managed to get access to correspondence and dig some documents and letters from the Camus and Monod families that no one had ever accessed before.
That sounds so cool.
So the book is fabulous. I totally recommend it.
Wonderful recommendations. I will put these books by François Jacob and also Sean Carroll on our website for our listeners to find for when they are in search of their next great read. And I know throughout our interview, we've talked about some of these different training opportunities you've had going between France and the United States. And I think the life of a scientist includes a lot of travel, whether it's to go to your new place of employment or to go to conferences, all these different opportunities to travel. So can you think back now, Franck, on all the places science has taken you and tell us about your favorite travel memory, where you were and why it was so memorable?
Yeah, I consider myself very lucky in that sense. Science and being a scientist is wonderful for many reasons, including traveling and meeting new people and becoming friends. And it's a wonderful experience. One of the very first trip that I took as a scientist, I was a master's student. I just spent the summer working on a project. In fact, the lab I ended up working in for my PhD, and I had some results. My mentor at that time, Henry Kennedy, said, why don't you go and present this preliminary result at a scientific meeting? You'll get great exposure and learn a lot. So I went to this meeting. I don't think those meetings exist anymore. I think they were called Summer Brain Research Meetings, something like that. This was in Amsterdam.
Gotcha.
And I was just fascinated. I distinctly remember two of the scientists that I later on studied, and I didn't really know their work yet. Pascal Rakish and Denis O'Leary, who are still very much active, very successful scientists, had these absolutely amazing debates. Debates like we don't see much in science anymore. They really were getting at it back and forth. And it was just wonderful to witness. And after that, they came to see my poster. I was a master's student. I was very, very impressed. They're both very different, but that left a long lasting memory. I still remember this, as you can tell. This was the first time I went to Amsterdam, beautiful city. And yeah, this was a great memory.
Well, it sounds like a great memory. It sounds both thrilling and terrifying. So what was going through your mind as you saw each one of these science heroes approaching you, standing at your poster?
Oh, I was stunned and my palms were sweaty and I was both miserable, but at the same time enchanted by the fact that they would even bother coming to see my poster. And they asked very specific questions. Some of them I couldn't answer.
That's normal.
I told them very honestly that I only worked on this project for like 3 months. But I think they were pleased, and I was certainly in heaven. I realized at that moment that the scientific community is amazing in that sense. Pascal Rakish at that time was very senior. I mean, he was probably 60 years old or something. And he was still incredibly passionate about science to the point of coming to see posters by young students. So I really enjoyed that.
Well, it sounds like a wonderful experience, like I said. And you mentioned you were in Amsterdam for this conference. Do you remember, did you have any time to do a little sightseeing outside of the conference?
Absolutely. And that's really the beauty of those meetings. This was like a summer school meeting. And so we had the organizers carved out sometimes for people to enjoy the city and go and visit. And I remember meeting a couple of other students at that time that I became friends with to this day. It's through those moments where you go and explore the environment that you forge those bonds, basically.
And I love that you mentioned the people. And I think definitely through travel opportunities, you get to meet wonderful people working in different universities, different academic environments. There are so many great people. And I think there are some stereotypes out there about what scientists are like. So we are always on a mission to break those stereotypes, to show the fun, funny, and just great side of scientists. So I love talking about some of these fun memories people have shared, some quirky traditions that spring up in laboratories. Do you have a story that shows the human side of science, Franck, that you can tell us today?
I totally agree with you. I think science is fun, and there are many different personalities in science. But at least personally, I was fortunate enough to be in different environments where people just like to have fun, maybe to compensate for the fact that science can be difficult sometimes.
Right.
I remember when I was a postdoc in Anirban Ghosh lab at Hopkins, Anirban started a game that was actually a lot of fun. We started playing this game where each member of the group would write something down, something funny or quirky or sometimes even borderline embarrassing that happened to us in the past. Then you would put this piece of paper secretly in a hat or something, and then someone would pick one of these at a time and then read it. And the game was to guess who wrote it in the audience.
That is awesome.
It's a very simple game, but if people play the game right, it's just a blast. I mean, it reveals really some of the fun stuff that people did or that happened to them. You just learn a lot about your colleagues and coworkers. And some of them are still my friends today, largely, I think, because I learned some traits, basically, that I would never have guessed otherwise. And when I say that to you right now, I'm struck by the fact that we're not playing this game anymore. And I think I'm going to start playing this game again in lab because I think it's actually a great game.
Franck, I think I'm going to steal it too. We're going to have to do this in our lab meetings as well. This is way better than your average icebreaker. So you don't have to implicate anybody, but what was the most surprising or strangest thing that you can remember that someone had written down on a piece of paper, but don't tell us who it belonged to?
Ooh, that's a tough one. I think somebody in the lab at that time said that they once stole a bike for what sounded like a fairly good reason, but they basically stole a bike, and they realized that the bike belonged to a campus cop where they were working. They returned the bike after learning that, basically. And I think this was a guy, he stole the bike because he got really late. He was supposed to have his new girlfriend at the time, something like that. It was something romantically involved.
Well, if he gave it back, then he just borrowed it without permission for some amount of time. So I like it. Well, fantastic game. And I love showcasing some of these exciting, fun, and funny things that scientists do in the laboratory, because I think you're absolutely right. You need some of these things to bring up morale, to have a little fun, release a little steam after you've been spending maybe long hours at a microscope or just long hours in the lab in general. And I know we've chatted a little bit about what you spend your long hours in the lab doing, some of the exciting questions that you're answering. But I want to talk big dream research questions here for a moment, Franck. If we gave you everything, if things like funding and staff and technology and feasibility were not a problem, what is the one question that you would want to answer?
I think the question I mentioned above is still a question I would love to contribute to. What is unique about the human brain? What allowed the emergence during evolution of human-specific cognitive abilities? Right? For example, what allowed some humans to start communicating through oral language first, and then soon after that, probably through written forms of language? Or even before that, what allowed humans in some obscure caves in Lascaux in France, for example, about 20,000 years ago, to start painting animals? Some of those paintings are absolutely remarkable, even though they were done more than 20,000 years ago.
Right. They're better than I could do now. So
That's right. Clearly, some of those paintings are trying to represent animals in movement. I found that fascinating. What allowed those early humans to start representing by drawing their environment? I think this is a giant leap in evolution. And through the study of these human-specific duplications and maybe some other genetic mechanisms, I really hope that our group will get some new insights into what makes our brain unique, and in particular, what aspect of brain connectivity might make us unique in that sense.
Well, this is a phenomenal question. Like I said, one of these ones that will keep you up at night thinking about what makes us unique, what makes our brains different, and how did they get that way? So great big picture question for us to think about, Franck. And we've talked a little bit during our interview about some of the people who've had a big impact on you. And I think these people are instrumental in getting you to where you are in your career. And oftentimes they get you there with some priceless advice. So can you share with us today, Franck, if you look back and thought about everyone who had an impact on you, who gave you the best piece of advice and what was it?
I can't really pinpoint to specific advice, but I distinctly remember that my postdoc advisor, Ara van Gogh, told me before I left his lab to keep my enthusiasm for science and to share that enthusiasm with students and coworkers. I've tried that, maybe not always very successfully, but I think overall I've tried to do this. And I think that if I ever lose this enthusiasm, I'll quit science and do something else. Because I think this job is fun, but challenging at the same time. And if you don't have that level of enthusiasm and this passion for science, quickly people notice it around you. And I think it's better to quit at that point.
Well, this theme of undiminished enthusiasm keeps popping up in our conversation. I like it. So is there any other last piece of advice, Franck, or any last note of inspiration that you want to end our interview on today?
Again, keep your enthusiasm high for science, and more generally, be curious, be critical. Keep the eye of a child when it comes to nature, biology, or science in general. I think it's so important in life in general. I think supporting science in any way you can. Science is one of the most exciting things one can do in life. I'm still convinced of that. So keep your enthusiasm high for science. It's a wonderful thing.
I love it. Very well said, Franck. And I think exploring your world, staying curious is something you can do whether you're a professional scientist or not. So listeners, take this advice to heart. And Franck, can you tell our listeners how they can reach out if they want to learn more about you and your work?
We've recently updated and sort of revamped our website. So it has a simple name. It's polleulab.com, so POLLEUXLAB.com. All our contacts are listed in there, including my email, so which is probably the best way to contact me by email.
And there are a lot of really cool pictures of neurons on there as well. So listeners, definitely check this out. Franck, thank you so much for joining us today and sharing a piece of your story with us.
It was a real pleasure, Mary. Thank you.
It was a pleasure to speak with you as well. And listeners, thank you as always for joining us. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guest favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.